Okay, so today we'll move into ethers, and then maybe a third of the way through we'll get into amines, and then on Wednesday we'll finish up amines, and then you'll have your Module 3 exam. Sometime Thursday or Friday, I'll submit your interim grades into SOAR, and then whenever they release that, you'll be able to see it. But you should download that Excel file to track your points and see where things are going. So we had left off last time talking about activating alcohols to sulfonates and then doing the SN2. One of the SN2 reactions that we learned in Organic 1 was the Williamson ether synthesis. So that's a pretty standard reaction, and that will lead us into the functional group ethers. So an ether has an oxygen atom that's sigma bonded to two different carbons. And so as we'll see, the carbon groups can be the same to form symmetric ethers. If they're different, the ethers are non-symmetric. And ethers are typically used as solvents in a lot of organic reactions. But what the reaction here is showing is that we have a tertiary alcohol reacting with a primary bromide via an SN2 reaction to make the ether. So strategically, any ether that you see can basically be made one of two ways. You're identifying the oxygen and then breaking the bond to either the carbon on the right or the carbon on the left. And that sets up these two different pairs of where the oxygen can come from, what alcohol, and then what the electrophile is going to be. So because it is SN2, you typically want your electrophilic carbon to be the less substituted one. So with that, we'll get into a little bit of the nomenclature and physical properties. So ethers, again, have that oxygen atom sigma bonded to carbons. And if the two groups are the same, then the ether is symmetric. If they're not the same, then it's non-symmetric, and that will kind of play out with how the carbon and proton data appear as well. So in the table here, we're going to run through the symmetric ethers based on carbon count. So each side of the oxygen, so the carbon chain length is going to be the same. If they're both methyl groups, the nomenclature is called dimethyl ether, and that would be the structure, and then you can see the molecular weight, the melting and boiling points. If we go up to a 2-carbon chain, so 4 total carbons, that's diethyl ether. And you see now this boiling point of about 35. That's right above room temp. And so diethyl ether is a common solvent used for reactions and extractive workups. Once we get beyond that, so the next one, dipropyl ether, this boiling point is fine for running reactions in, but it's not used commonly for extractions during workup because it's a bit too high. And then from there, we can get branched ethers as well, where the side chain has branching. And then we can just continue on up in carbon count. The last one is diphenyl ether. So each chain of the ether is a benzene ring. And if you look at that, that's a pretty high boiling point. So sometimes when you need to heat the crap out of a reaction, you would use a solvent like that where you can really crank it up. And as I said, ethers are commonly used as solvents because they're inert. So diphenyl ether, there's not much that's going to react with it. So it's inert as a solvent, and then it has that high boiling point that you can get, if we use the mark for water at 100 degrees, you know how hot that is. So 259, we're really cooking it there. And again, that all plays back into, the Arrhenius equation and heats of energy of activation for transition states. So when the ether is symmetric, you just identify what the chains are, and you call it dialkyl ether. If it's non-symmetric, then we're identifying the carbon atom with the chain with the smallest number of carbons, putting oxy at the end, and then using the longer chain as the parent. So for this linear case, the longest chain is 2 carbons, so that's the parent ethane. And then we're going to use the 1 carbon chain as a substituent, so methoxyethane. And you can see that logic play out through the 1-methoxypropane, 2-ethoxypropane. And those are all cases where that oxygen is at the end of the carbon chain. If the oxygen is internal, it's then branched. And so when it's branched, you have to specify the location of where it's bonded. So we know propane's 3 carbons. The methoxy group is there, and the 2 is just stating it's at position 2 on the chain. So the substituents on oxygen can also be rings. And really what you're doing now is just comparing the size. So this side has 6 carbons. That has 1. So whatever has the most number of carbons becomes the parent. So methoxycyclohexane. Then in here we have a 6 versus a 3. So the 3 is named as the substituent and the 6 is the parent. So that's how you name them. It's not the most exciting stuff in the world. The 2 chains of the ether can be constrained or tethered together. That forms a ring. And here are some of the ring sizes from 3 to 8. What's unique about these is they're typically just given common names, so they're not IUPAC names. So the 3-membered ring is called an epoxide. So those are special ethers because of the strain that makes them reactive. We'll talk about a reaction or 2 of them. Oxytanes, we're now up to 4. Those have some ring strain present as well, but not as much as the epoxide. THF, tetrahydrofuran, that's a typical organic solvent. So I like to think about, just want 2. So this was diethyl ether. So as I said, diethyl ether is a nice solvent. It's acyclic, so there's a lot of rotation that can happen amongst those sigma bonds. So we can imagine THF, if you take these terminal carbon atoms, bond them together, you get a ring. And so that constrains the rotation. It makes it a little bit more polar. It's used commonly to help dissolve things that have a little bit more ionic character to them. Tetrahydropyran, the 6-membered ring, we're going to see that when we get to biomolecules and we start talking about carbohydrates. So a carbohydrate can have this form, and then each carbon atom is basically has an OH group or something. So that's what makes carbohydrates special. 7-membered rings are oxypanes, and 8 are oxocanes. So these are probably less commonly used in lab, but they're seen quite a bit in natural products, so molecules isolated from plants. In terms of the spectroscopy, or how do we know an ether is in a molecule, for IR, mass spec, proton, and carbon, you'll notice that all of these are in yellow, which means there's nothing with a high level of confidence that is a diagnostic signature. For IR, a carbon-oxygen sigma bond is in the fingerprint region, and since that has a lot of stuff, it's hard to pull out as okay, that's an ether. Mass spec, you may see a fragment where the parent minus 18, where that corresponds to water, could be a daughter ion. Proton NMR, when you see signals between 3 and 5, that's an indicator that that carbon atom has an oxygen bonded to it. And then the same thing for C13, so that H should not be there. So ethers are one of the more challenging functional groups to figure out through spectroscopy. So let me just use the diethyl ether as an example. So we know that oxygen has 2 lone pairs. So the oxygens delta minus, those carbons are therefore delta plus. So the molecule is going to have a permanent molecular dipole based on those individual polar covalent bonds. So the lone pair on oxygen makes it a hydrogen bond acceptor only. So to be a donor, you have to have OH or NH. So to a certain extent, if you had a beaker of ether and then a beaker of water, you're going to get some mixing or miscibility. As the side chain, as the length of these carbon side chains increase, that solubility or miscibility is going to decrease. So diethyl ether, and water can hydrogen bond like that. So the ether is the acceptor, the water is the donor. So through that interaction, you'll get a little bit of miscibility. So have you used a separatory funnel in lab? So when you pour water in there, you pour ether, you're going to see a phase split, but there's still a little bit of that ether that goes into the water. Because ethers do not have an OH group, they'll typically have lower boiling points than the corresponding alcohols of the same carbon count. And they're useful for as solvents because they're inert. So if we look at the unique sets of hydrogens, what functional groups they belong to, what those pKa's are, the hydrogens are usually not very acidic, so that makes them compatible with these reactions that are strongly acidic or basic. And here's a list. So diethyl ether, cyclopentyl methyl ether, THF and methyl THF are common ones that are used. So to that first slide I talked about, if we look at the ether that's shown here, 1-ethoxy, 1-methyl cyclohexane. If we had to make that, we could generate 2 pairs of reagents where one of them would include the oxygen. That would be a nucleophile. Then the other one has to have a leaving group, that's the electrophile. The rational way to do this, once you identify the oxygen, you look at each carbon atom. Then you classify that as if it were an alcohol. So the one on the left, we would say that's primary. The one on the right is secondary. So with the green arrow, that would be the preferred bond to break in terms of coming up with a pair of reagents to make that molecule. So when we do that, we see that primary carbon, if we put a leaving group on it, generates a tertiary alcohol of this form. That's the alkoxide. And that's good for SN2 because that's the less substituted electrophile. The alternative way shown in red, the reason that's not preferred, because now the leaving group is on a tertiary carbon. And so for SN2, that's not favorable. And under these conditions, can anyone think of what functional group product would probably be formed if you ran this reaction? What other mechanism do you know? So instead of doing substitution, this would do an elimination. And so specifically with tertiary leaving group, instead of acting as a nucleophile, this would now be a base. And so it would do an E2 elimination. And that could form, say that as a major or this as a minor, depending upon, things like concentration, heat, that sort of thing. But since it's elimination, you would end up with alkenes in this case. So that's what you need to think about to make ethers like that. As I said, when we saw the ethers that were constrained in rings, the epoxide or three-membered ring ether is pretty special because there's a lot of strain in that ring. So we have sp3 hybridized atoms, and the bond angle should be 109, but it's less than that, so we've put a lot of angle strain in there. Essentially, what that is doing is making these CO bonds very weak. The ring wants to open up to release that strain. The reagent shown on the slide here, so MCPBA is metachloro should be peroxybenzoic, but so metachloroperoxybenzoic acid. The per or peroxy part is this second oxygen, which I've highlighted in blue. You're normally used to seeing a carboxylic acid as CO2 and then H. An oxygen-oxygen sigma bond is called a peroxide. And since we're ending up with OH here, it's a peroxy acid. And that makes that oxygen, if we think about it, let me ask you a question. We're starting with an alkene, MCPBA, and that blue oxygen ends up as the epoxide. So between the alkene and this, which one is the nucleophile? Which one supplies electrons? So that pi bond is, compared to a sigma bond, it's weaker. So the electrons are going to come from that pi bond, so this is a nucleophile. And so this oxygen is electrophilic. And what is happening is, of the two carbons of the alkene are simultaneously bonding to that oxygen. So the hybridization changes, and then you get a sigma bond to the same oxygen. So it's a 1-2 addition. And the way that it happens is at the same time. So in the plane of that alkene, the oxygen comes from one face, and those two bonds form at the same time. So that's a syn and then synchronous. So that's an easy way to make epoxides, meaning that anytime you see an epoxide, you can think backwards and say, okay, that carbon and that carbon can come from an alkene. So that little arrow in the middle there, that means retrosynthesis. So if I see an epoxide in my product, what can it come from? So those two carbons, and I just do a swap from epoxide to alkene. If we take that one step further, where do alkenes come from? We can make alkenes through elimination. So epoxides will exhibit this amphiphilic reactivity, and what I mean by that is under acidic media they'll act one way, and then under basic or nucleophilic media they act a different way. Let's first consider that epoxide under acidic medium. And what I want to do is look at both carbon atoms of the epoxide and classify them like they were alcohols. So this one would be primary, that one would be secondary. So if we were under conditions with hydronium ion, hydronium ion's an acid, so this has to be a base. So that oxygen would be protonated. Now the oxygen can't leave the molecule, but we can break a carbon-oxygen sigma bond. And so we can do that either of two ways. So say I break open the one from the primary, and that now generates a primary carbocation. Or if I do it the other way, that now generates a secondary carbocation. So in acidic medium, the preferred way of breaking the bond is the one that generates the more substituted carbocation. And so depending on what else is in there, that carbocation will get trapped by a nucleophile. If we took that same epoxide and now we were under a different set of conditions, say we had potassium cyanide with acetone and heat. Potassium cyanide is a strong nucleophile, so this is going to react through an SN2 mechanism. So cyanide is the nucleophile. One of these carbons is going to be the electrophile. Because it's SN2, we now want to use the least substituted carbon, so the primary, meaning that these electrons on the cyanide would attack that carbon to pop the bond open that way. And so really what the point of this is, whether it's acidic or basic media, the epoxide oxygen is going to end up on a different carbon. Questions? So one last reactions of ethers. I said ethers are pretty inert, but they can undergo an SN2 reaction if the conditions are primed for that. And in the game of organic synthesis, we call this a deprotection. So ethers are pretty stable, and if the substrate and the conditions allow for it, you can remove one of those side chains. So if we put our lone pairs on that oxygen, if we treat that ether with HBr, which is a strong acid, we're going to protonate that oxygen. So that's an acid-base reaction. Oxygens plus, we've generated Br minus, that's now an anion. And what can happen because this oxygen is activated, the bromine Br minus can act as a nucleophile through an SN2 reaction. If we classify the hybridization of those carbons, the blue one is sp2. The green one is sp3. An SN2 reaction must have the leaving group bearing an sp3 hybridized carbon. So in the choice of where that attacks, it has to be the green one, because that blue one is not a good electrophile to break that CO bond. And when that happens, we end up with phenol as the alcohol and bromoethane as the second product there. So really, what is this? if you took these two together, phenol and bromoethane, that's what you would use to make the ether. This is just showing you how you could deprotect or remove that side chain. So what is the potential issue with this? This system's kind of rigged in that reaction can only take place at one carbon. If you have another carbon here that's sp3 hybridized, you're going to end up with a mixture of stuff, and typically you don't want that. So this reaction has a narrow set of applying it in real life. So this is the last slide for the ether alcohol part. And just to say that I checked the box, this is on thiols. So on the periodic table here, in this group, there's oxygen, sulfur's right below. So pretty much everything we've learned so far for alcohols, it applies to thiols, which are SHs, and then sulfides are the sulfur equivalent of ethers. So I have my, let me say that's, I don't know, not 2/3 full of water here, H2O. It's definitely not filled with H2S because that's a poison. And it's interesting because, again, sulfur is just right below oxygen, but the difference in electron configuration and size and all that makes it a totally different molecule. So hydrogen sulfide, that's the water equivalent. If you take your normal alcohol and just replace the O with sulfur, those are called thiols or mercaptans. So they have the SH group. Has anyone ever smelled a thiol? Typically when you're driving down the highway and you're like, it smells like skunk, that's a thiol that you're smelling. So they smell pretty bad. If you continue that logic on, so below sulfur is selenium. So these smell, they smell like, a truck stop bathroom. But selenium compounds will physically make you gag or throw up. They will elicit that response. They're so bad. So the nomenclature is, you just append the word thiol at the end, and then depending on where it is, you get the different classes, just like your normal alcohols. One consequence of the change from OH to SH3 is sulfur is a little bit bigger than oxygen, so it doesn't affect the physical properties as much because it's not a good hydrogen bond donor acceptor as OH is. So that would be the sulfur equivalent of an ether. Those are called sulfides. And then you could make them with the same sort of strategy as the regular ethers. Any questions before we move on to amines? So the way that the chapters are organized in how the functional groups lay in those different chapters. I'm going to introduce amines in a couple reactions. How do you get nitrogen into a molecule? There's more reactions, but those are saved for the next module because it better fits with how the nitrogen's introduced at that point. So for the rest of today and Wednesday, we're going to talk about structure, the nomenclature, the physical properties, spectroscopy, why are these things relevant, and then probably on Wednesday we'll get more of those initial reactions, but these are not all of the reactions for amines that we're going to see. So amines have 4 classes. Remember, alcohols only have 3. Amines can be primary, secondary, tertiary, or quaternary. The difference is you're looking at the number of carbon substituents now bonded to the nitrogen. With alcohols, you are looking at the carbon bearing the OH group. So let's run through a couple of these classes. So the first 3, we're going to increase the carbon count so that we can change the class. So with 1 carbon, that forms methylamine. Two carbons, if I have them both as methyl groups, that's dimethylamine. 3 would be trimethylamine. The difference between primary, secondary, and tertiary, if you look at the boiling points, this isn't going to be the strongest claim known to man, but primary and secondary can be hydrogen bond donor acceptors. The tertiary can only be an acceptor. So let's look at this carbon count 2 and then this 2 here. So in this second one, I've just used both carbons to make a longer chain, but now it's a primary amine, so ethylamine. So depending on your carbon count, you can generate these different isomers and make different classes. And so whether you end up with NH2, NH, or just N, again, that's going to affect the physical properties. So of all of these, I would say this one here, triethylamine. This is a common organic base that's used. So we'll talk about amine basicity. So because it is a tertiary amine, the nitrogen has a lone pair. A lone pair can act as a base to abstract hydrogens. And then so we need to talk about what is the strength of this base? What sort of hydrogens can it abstract from different functional groups? So I'm just drawing a general sort of bonding pattern. where the nitrogen has 1, 2, 3 sigma bonds and then that lone pair. Down in the structures, that lone pair is represented by this gray orbital. So you can think of that as being an sp3 hybrid orbital. Because it's not an actual substituent, the nitrogen is not tetrahedral. All the lone pair is doing is kind of shoving those 3 other groups into a plane. And we would say the geometry is trigonal pyramidal. So the lone pair isn't really counted as a substituent. So we give it this other name for the geometry. So ammonia NH3. So of our sort of main elements, carbon, nitrogen, oxygen, ammonia is one of the fundamental molecules. If we start substituting H's for carbons, so here we've made methylamine, that's primary. We do a second substitution, that's dimethylamine, it's now secondary. The third, trimethylamine, that's tertiary. Since that lone pair is available for bonding, it can form a 4th bond. Because the nitrogen no longer is neutral, it has 1, 2, 3, 4 sigma bonds. It ends up with a positive formal charge. And that would be balanced off by some anion X minus. And so overall, these are referred to as salts because it has that form of something plus, something minus. And because now there's that sigma bond, the geometry of that nitrogen switches to tetrahedral now. If you've ever taken a prescription drug, if you look at the active ingredient, sometimes, they'll have this form like drug name dot something like citric acid or something. What that's telling you is that within that drug, there's a nitrogen that's been protonated by citric acid. And so that the anion is citrate. And the reason that is done is because typically when you think of like salt, you think of solid. You think of something with, a high melting point, a nice solubility in water. So amines, depending on the structure of the molecule, electronics, all that stuff, if you want to sell a drug, you want it to be a solid that you can handle, you can put in the little pill, either the capsule or whatever, so you can measure it out easier. So making the salt of something is a lot easier to manufacture and make the required dose. And again, it gives it those desirable physical properties. One thing that is unique to nitrogen due to that trigonal pyramidal geometry, if you look at the structure shown in the middle there, the nitrogen atom has 3 different substituents. You have methyl, ethyl, and propyl. Then you have your lone pair. It turns out you could draw a mirror image of that, which is shown to the right. And so those are actually enantiomers of one another. But really what is happening is just you're flip-flopping that lone pair from one side to the next. And that's called an inversion. And it happens pretty fast. So even though these are, in theory, enantiomers, you can't really isolate either one in the sense of, okay, through some means I got the one on the left, I take an optical rotation, it's going to have some specific rotation, then the one on the right has the equal magnitude but opposite sign. This happens so fast that it's hard to determine that. If these groups get sufficiently bulky, then in theory you could freeze out one of these to prefer one stereoisomer over the other. But overall, this is called a Walden inversion or umbrella. So if it's, a normal umbrella, the rain's coming down, it's washing over, keeping you dry. But if it's windy and you hold the umbrella, to the direction that flips it, now the umbrella looks like a bowl and it starts collecting water. That's essentially what these amines do. So that the stick of your umbrella is like this lone pair. Notice now when we get up to these secondary amines, they don't have a separate name. When we took the H of an alcohol and put a carbon on there, we were calling that an ether. If we have a secondary amine, we can tether groups together to make rings, and these are called heterocycles. Notice that all of these are at least secondary. We could substitute H to make them tertiary. But just like with oxygen, they have these special names as shown on the slide. The 3-membered ring is called a xeridine. And even though there's ring strain, just like with epoxide, these are not as reactive as epoxides. So azetidine, pyrrolidine, piperidine, azepane, and oxocane are the common names. So now I want to talk about aromatic amines, like everything before the substituents bonded to the carbon, all of the carbons were sp3 hybridized. So for these, what I'm going to classify them as at least one of the carbons is sp2 and it's further constrained into a ring. So that's what I mean by aromatic rings. The first one is called aniline. So we would classify this as a primary amine. We have an sp2 hybridized carbon, and that's in a benzene ring, so aniline. Then we can substitute H for methyl. That makes N-methyl aniline. So anytime you see this nomenclature with this capital N, whatever comes after it, that group is bonded to the nitrogen. So N-methyl and then anilin's the parent. Then the last substitution, notice now NN, there's two N's, then dimethyl. And then aniline is the parent. So the names and physical properties are shown in the table here. Look at the boiling points for all of these. They're pretty high. That's due, again, to hydrogen bond donating and accepting. The other thing is that aromatic ring, we know from looking at it, every carbon is sp2 hybridized. So it's trigonal planar. The ring is flat. Essentially what happens is if you zoom in, you have a flat ring, you have another one. These things just stack on top of each other. So we've talked about hydrogen bond donors, acceptors. So pi-pi stacking is another mode of non-covalent interaction that affects physical properties. And if you work in a lab long enough, you'll see that when you start appending these rings, the molecules, we start calling them bricky. So as you get more rings, they're less soluble in things. It's harder to get solvents to dissolve them. The melting points get really high, and that's due to this pi-pi stacking. And we'll revisit that concept again when we start talking about the structure of DNA, how the heterocyclic bases are stacked on the DNA strands. Let me point out, the nitrogen is bonded to the carbon of a ring. So in the next series, we're going to stick that nitrogen inside the ring. So again, and then make the ring aromatic. So in our first two modules, we talked about conjugation and aromaticity. So all of the amines shown on this slide are aromatic amines. So they obey the Huckel's rule, the Frost circle, those things we talked about. So pyrrole is pretty common, pyridine, quinoline, and isoquinoline. And you'll notice they all have pretty high boiling points. What I want to kind of refresh our memory on the pyridine, quinoline, and isoquinoline. I drew in the lone pair. Nitrogen is sp2 hybridized. We see the pi bond right there. So the nitrogen p orbital and that carbon p orbital form the pi bond, meaning that this lone pair exists in an sp2 hybrid orbital. In pyrrole, the nitrogen is also sp2 hybridized, but if you count the number of sigma bonds, 1, 2, 3, and then we draw in that lone pair, this lone pair has to reside now in a p orbital. So why is that important? One of the reactivity profiles for amines, I said, they're basic. The nitrogen atom lone pair will abstract an acidic hydrogen. To abstract a hydrogen, the lone pair has to be available to do that. So pyridine, quinoline, and isoquinoline, those are fine to use as bases. Pyrrole is not a good base because that lone pair is in a p orbital that's tied up in the conjugation to make this aromatic, meaning it's not available to act as a base. So in the series of primary to secondary to tertiary, again, for primary, there's only one carbon group. So that means there's NH2. For secondary, there's NH. And then tertiary, there's no more H's bonded to the nitrogen. So the primary and secondary, those can be hydrogen bond donors and acceptors. The tertiary can only be an acceptor. And so again, that plays into how the IRs look in terms of NH stretching, other physical properties like miscibility in water. And the other thing I want to point out so in blue, primary and secondary have an NH. So those hydrogens can be acidic, meaning they can be deprotonated. But if we look at the pKa value here, those are not very strong acids. You know, if we compared that to say methanol, the OH group has a pK of about 16. This amine is 35. So that's not a very strong acid. And that all gets back into looking at when you do deprotonate it and you look at what's formed, how does that species stabilize the negative charge? So realize this is not the best drawing in the world. The compound shown is methylamine, and then this stick right here, that's the lone pair. So of the methylamine, that NH, that can be a donor for water to accept. then this lone pair of the nitrogen can accept an OH from water. And so through those interactions, we can make some claims about miscibility in water and other polar or non-polar solvents. So a couple things, I believe, I've really highlighted the case of the primary amine, and when it gets into secondary and tertiary, it's a little bit more difficult to use the spectroscopic techniques to sort of classify these things. For a primary amine that has NH2, I would say most of the time in that region 1, because there's 2 hydrogens, you're going to see a little doublet looking thing that indicates it's a primary amine. For all of the cases, primary, secondary, tertiary, as long as there's an odd number of nitrogens, the parent ion mass will be odd. Just like with ethers, the proton and carbon, these are in yellow because they're not terribly indicative that you have that specific NH2, NH, or N. But if you see some signals that are shifted further away from 0, okay, there's at least a heteroatom there. Then I have to look at these other things to help narrow it down. Look at this. So here's our little doublet thing I was talking about. So this is region 1. If you look at trace that, it goes down, then back up, down. So that's what I mean by a doublet. If you remember what an alcohol looks like, remember an alcohol goes down pretty far and then back up. A carboxylic acid sort of merges more over here. So that is a unique signature for NH2. And then as you move up in the class, so this structure over here now is dibutylamine, so it's secondary. There's only one NH, and there's this little blip, which is not terribly diagnostic of saying, okay, I'm 100% confident that is a secondary amine. So the last case was tertiary. So this is tributylamine. Notice there's nothing here. When you look at this, there's no carbonyl. And so really the IR is not telling you a whole lot about the presence of an amine. You'd have to look at the mass spec data to get the information. Okay, there's one nitrogen, then you'd have to look at the carbon and the proton to start trying to figure out exactly what all of those substituents on the 3 different things of that amine are. So here's aniline. And this signal here at 93, that was the parent ion. So because it's odd, we can assume that there's going to be an odd number of nitrogens. That doesn't immediately tell you, is it 1, 3, or 5? You just kind of have to work through the scenarios. Say, okay, let me start with 1 nitrogen, which is 14. So I could take 93 minus 14, try to estimate a carbon H count, see what kind of works out. Does it match with the carbon and proton data? If not, then you can go up to 3 nitrogens, do that sort of math as well. So we have a couple more minutes, and I'll just show you some relevance of these nitrogen-containing compounds. So a lot of, I would say, organic chemistry has evolved, through human interaction with plants. Like, why do we eat things? So through time, we've got to the point where, okay, we can pretty much identify what is in that plant. So whatever those molecules are, they're just given this general name, natural products. So molecules that contain nitrogen are further classified as alkaloids. And in the classical literature from 100 years ago or whatever, a lot of these papers would start off with bitter constituents of whatever plant someone was interested in. So if you think about that, that title, bitter means something you taste, right? So IRs, NMRs, those things didn't exist. And so what did people do? They would take the leaf and start chewing on it, as your tongue and taste is a chemical sensor. And so as enough of this knowledge became known, it would say, okay, this leaf tastes bitter, so therefore I'm sort of confident there's an alkaloid in there. And then using all these principles about physical properties, you could take a bushel of these leaves, soak it in some solvent, and start extracting things out, running some TLCs to figure out, okay, of that, how many spots do I see? So the reason nitrogen is special is because that bonding pattern of 3 sigma bonds, that lone pair, give it this pharmacological property that makes it bioactive, in the sense that when it's taken, it elicits some response in the body. So a class of alkaloids called the tropane have that substructure. So this molecule to the left is called tropane, and then this molecule to the right is called cocaine. And if you look at what are the modifications that have been done, so I'll highlight those in blue. So there's basically 2 different types of esters on that tropane skeleton that make it cocaine. And so why is that interesting? Why is it controversial? Cocaine is a natural product. I mean, plants make this. But as humans and society, it's illegal, right? So if something's natural, how can it be illegal? So we have these agreements with each other, with laws and that sort of thing. But cocaine used to be used as an anesthetic specifically for, like, if you had a toothache, you'd rub some of the booger sugar in there, and it would alleviate that pain, right? They say it used to be in Coca-Cola as well. Man, those were the days, right? But now it's illegal, and think about all the implications of that. All the downstream societal effects. So what we'll continue on with Wednesday is a little bit more of these, then we'll get into some of the reactions, and we'll finish up that module.